Welcome to medicinal biochemistry class on nucleic acid metabolism. Okay, so today we will put an insight into what are these nucleic acids and what is their biological role. So he is Friedrich Meissner and she's Rosalind Franklin. So these two scientists have great contribution in the discovery of nucleic acids. Okay, so in your future classes and today's class, we will look into that, like what is their contribution. In fact, there are many scientists who have contributed by large for the development of this science of these nucleic acids, and they have a major role, and they have given a lot of breakthroughs in medicine, that is in drug discovery. So we'll go through a brief history about how this discovery of nucleic acids started. So nucleic acids were first discovered in 1868 by 24-year-old Swiss physician Friedrich Meissner. So he isolated a new compound from a nuclei of WBCs, that is white blood cells. So the compound was neither protein nor lipid nor carbohydrate. So the three important components of our food and which provide energy for us, and they are the building blocks of our body. Okay, so this the new compound that Richard made, that is Friedrich Meissner, who isolated from the nuclei of WBCs, was neither of these three. So that was totally altogether an compound with a new chemistry. So this novel type of biological compound he named as nuclein because he isolated it from nuclei of WBCs. So today his discovery is known as DNA. That is, in fact, he isolated DNA for the first time from WBCs. So along with DNA, so there are other major type of nucleic acids in the cell. So that is one more major type is RNA, that is ribonucleic acid. Okay, so that was a brief history about these nucleic acids. So today, at the end of this class, you will be able to define water hydrogen bond. So what is the, and define water hydrogen bond donors, define hydrogen bond acceptors, define purines and pyrimidines, define nitrogen bases, define nucleosides and nucleotides, define nucleic acids, and recall significance of nucleic acids. Okay, so these are today's learning objectives. So this is a brief introduction about nucleic acids. So this class will help you to study the metabolism of these nucleic acids, the structure of DNA, structure of RNA very well. So this will be the basis for the future classes. So now what is a hydrogen bond? Okay, so let us take the example of water. So this is the structure of water. So you know that water is the molecular weight of water is very less, that is, it is a very small molecule. It is having a molecular weight of 16 Daltons. So in spite of that, so it exists in the form of a liquid at a room temperature. Its boiling point is quite high, that is 100 degrees Celsius. Okay, so the reason for that, when compared, that is the higher boiling point. So the reason for the higher boiling point of water, when compared to its low molecular weight, is because of its ability to form hydrogen bond. So we look into that, how this water molecule forms a hydrogen bond and what is this hydrogen bond. Okay, so water, as you know, that it is a highly polar molecule. So it is having two hydrogen bonded to oxygen atom. Oxygen is a highly electron withdrawing group, and it is having two lone pair of electrons. Okay, it is having two lone pair of electrons. So the hydrogens are electron releasing group and oxygen is a highly electron withdrawing group. So due to that, what happens is, so oxygen will carry, and that is oxygen in water will carry an partial negative charge, and hydrogen will be carrying partial positive charge. In fact, the positive charge will be very high and the negative charge will be very high because of large difference in the electronegativity between these two atoms. Okay. So here, so when one molecule of water comes across one more molecule of water, that is when one more molecule of water comes near to this water molecule, what happens is, so the lone pair of electrons in the oxygen atom will play an important role here. So here what happens is, so the positive charge on one of the hydrogens of water molecule will get attracted towards the negative charge on the oxygen of the another water molecule. Okay, so due to this, what happens, an electrostatic force will hold these two water molecules, and the lone pair of electrons on the oxygen atom will contribute to form a weak bond, what we call it as hydrogen bond. In fact, so this bonding or this electrostatic force of attraction acting between these two molecules is very weak. It is weaker than covalent bond, it is weaker than ionic bond. So when you consider it as a single bond, but when there are millions of such linkages, okay, when there are millions of such linkages between many molecules of water, what happens is it creates an astonishing amount of stability. Okay, so due to this, the boiling point of water is very high, and water is a liquid at a room temperature, and it is not a gas because it is stabilized by these hydrogen bonds. Okay, so now we will define what are these hydrogen bonds. A hydrogen bond is a special kind of dipole-dipole interaction that occurs between a hydrogen that is bonded to an oxygen, a nitrogen, a fluorine, and the lone pair of electrons of oxygen, nitrogen, or fluorine bonded to a hydrogen in another molecule. So here, so what is an hydrogen bond? It is the dipole-dipole interaction between two molecules. So the condition here is the two molecules should have hydrogen bonded to most highest, that is elements with highest electronegativity. So these oxygen, nitrogen, and fluorine are the three elements with the highest electronegativity. Due to that, what happens is the positive charge on the hydrogen and the positive charge on the electronegative atom will be very high. So when two such molecules come across, they form hydrogen bonds. Okay, so that is a hydrogen bond. So it is a kind of dipole-dipole interaction that occurs between hydrogen that is bonded to an oxygen, nitrogen, and fluorine, and the lone pair of electrons of oxygen, nitrogen, and fluorine bonded to a hydrogen in another molecule. Okay. So the next thing is, so there are two parts in these molecules which can form hydrogen bonds. That is, one is hydrogen bond donor and another one is hydrogen bond acceptor. Okay, so here a hydrogen bond donor means, so the molecule which contributes hydrogen in the hydrogen bonding. Okay, the molecule which contributes hydrogen in the hydrogen bonding. Hydrogen bond acceptors means the molecule or the molecule which contributes electrons, that is the lone pair of electrons in the hydrogen bonding. So the donor in an hydrogen bond is usually a strong electronegative atom such as nitrogen, oxygen, or fluorine that is covalently bonded to a hydrogen. Okay, so the hydrogen acceptor is an electronegative atom of a neighboring molecule or ion that contains the lone pair of electrons that participate in the hydrogen bond. So these are the examples of hydrogen bond. So this is nitrogen. So in fact, it is ammonia, NH3. So it is carrying a lone pair of electrons. Okay, sorry, let me clean this. Okay. So this is ammonia, NH3. So here this is forming an hydrogen bond with one more molecule of NH3, that is ammonia. So here this element nitrogen is a hydrogen bond acceptor because it is contributing lone pair of electron, and this nitrogen is a hydrogen bond donor because it is contributing the proton or hydrogen to form the hydrogen bond. Okay, so here ammonia is forming hydrogen bond with water molecule. So here ammonia is hydrogen bond acceptor and water molecule is the hydrogen bond donor. Okay, similarly you have hydrogen bonds between hydrogen fluoride, and so this are the hydrogen bonding structures. So this is in fact 2D structure of water molecule forming hydrogen bond with its own another water molecule. Okay, so what is the biological role or biological significance of these hydrogen bonds? Why are we studying this hydrogen bond before we study nucleic acids? Because so the two strands of double helix in DNA are held together by hydrogen bonds. So it plays an important role in the structure of nucleic acids. So here in DNA the double strands are held together each other by hydrogen bonds. So the intramolecular hydrogen bonds is partly responsible for secondary, tertiary and quaternary structure of proteins and nucleic acids. Okay, so these hydrogen bonds will help them to form and maintain the specific shape that is required for the molecule so that it can support the life. Okay, so now we will see some a brief chemistry about the nucleosides, the building blocks of these nucleic acids. Okay, so there are two chemical structures or the compounds which are the building blocks of these nucleic acids, that is purines and pyrimidines. So we'll look into that one by one. So first we'll begin with pyrimidine. So pyrimidine is a 6-membered aromatic ring structure. It has a 6-membered aromatic ring structure in which so there are 2 nitrogens in the ring at 1, 3 position. So that is why, so this is an heterocyclic ring. What is an heterocyclic ring? So where in the cyclic ring structure carbons are replaced by other elements such as nitrogen, oxygen, sulfur. Okay, so here the carbons are replaced by nitrogen at 1, 3 position in a benzene ring. So this is pyrimidine. So it is a 6-membered heterocyclic compound with the 2 nitrogen atoms in the ring, and it has nitrogen atom at positions 1 and 3 in the ring. So purines are fused ring heterocyclic compounds. So here the pyrimidine ring is fused with an 5-membered nitrogen containing ring, what we call it as imidazole. Imidazole is a 5-membered heterocyclic compound where the nitrogen is present at 1, 3 position. So here pyrimidine is fused with an imidazole ring, so that is called as a purine. Okay, so purine is a heterocyclic aromatic organic compound that consists of 2 rings, that is pyrimidine and imidazole fused together. Okay, so now what are nitrogenous bases? Okay, so here these are also called as nucleobases, or simply when we are studying nucleic acid we simply refer them as bases. Okay, so these are nitrogen containing biological compounds that form nucleosides, which are further converted to nucleotides, and nucleotides are the building blocks of nucleic acids, that is DNA and RNA. Okay, so these nucleotides constitute, or they consist of, that is they constitute the basic building block of nucleic acids. Okay, so now, so what are the nitrogenous bases of DNA and RNA? So there are 5 nitrogenous bases of DNA and RNA, that is adenine, guanine, cytosine, thymine and uracil. So in DNA, So it is adenine, guanine, cytosine and thiamine, and in RNA it is adenine, guanine, cytosine and uracil are present. So the difference between DNA and RNA, the components, that is the difference in the content of nitrogen bases between DNA and RNA is thiamine and uracil. Thiamine is present in DNA, and in place of thiamine, uracil is present in RNA. Okay. So now we'll define this. What are these nucleosides and nucleotides? Okay. So here, so if you look at these structures, so the nucleoside means, so it is nitrogenous bases plus ribose sugar attached to it. Okay. So that is called as a nucleoside. Okay. So this is an example of deoxyadenosine. So why it is called deoxy means because here at the second position oxygen is removed. Okay. So in the sugar molecule, that is in the ribose sugar, at the second position oxygen is removed. So that is why it is called as deoxy. So this is a nucleoside, and when you add phosphate group to this, triphosphate group to this, okay, so then it is called as nucleotide. Okay. So this is the structure of DATP, that is deoxy ATP, because here the oxygen is removed, so it is denoted as deoxy ATP, adenosine 5-triphosphate. So this is a nucleotide and this is nucleoside. So here a nucleoside means nitrogen base plus ribose. Nucleotide means nitrogen base plus ribose plus phosphate molecule. Okay. So the next is that is the structure of nucleotides. So the structure of nucleotides, in the structure, so there are three major components. One is nitrogen base. It can be adenine, guanine, cytosine, thiamine or uracil. So and the ribose sugar, and at the fifth position the phosphate group. Okay. So here, depending upon the presence and absence of oxygen at the second carbon atom, so it is called as a deoxyribose nucleic acid or simply ribose nucleic acid. If oxygen is absent, then it is called as a deoxyribose nucleic acid. If oxygen is present, it is called as ribose nucleic acid. Okay. So now what is a nucleic acid? So nucleic acids are biopolymers. So it is built by nucleotides. Okay. So these are biopolymers composed of nucleotides as monomers. Okay. So these are very essential for all known forms of life. Okay. So there are two main nucleic acids, two main classes. One is DNA and another one is RNA. Okay. So here, most of the times these nucleic acids are considered to be, or nucleotides are considered to be non-essential molecules because they are synthesized inside our body. But that is not most of the times. These the quantity that is synthesized inside our body is not sufficient, and when it is consumed in the form of food, so it is consumed in the form of nucleoprotein. So that is in the stomach, it is broken down into nucleic acid and a protein. Protein will undergo metabolism as we have discussed in your previous class, and nucleic acid will break down in, and it will release many nucleotides, and these nucleotides will break down further and they'll form nucleoside and phosphate, and this phosphate will further breaks down and it will release the nitrogenous base and ribose sugar, and these can be utilized again for the biosynthesis of nucleotides and then to nucleic acid as per our requirement. What is the significance of these nucleotides? Okay, so what is their role? So these are the precursors or building blocks for DNA and RNA. So these are the precursors for DNA and RNA synthesis. So these are essential carriers of chemical energy like ATPs and nucleotide. Okay, so that is, we call ATP as an what? Energy currency of cell. Okay, so the components of the cofactors, that is NAD and FAD and coenzyme. So these are the components of the cofactors. Okay, and further, so the UDP glucose and the CDP diacyl glucose are activated by these nucleotides. So cyclic AMP, cyclic GMP are also the second messengers. So these molecules play a very important role in regulation of many metabolic pathways. So what are the sources of nucleotides? So there are two major sources of nucleotides. One is salvage pathway, so where our body utilizes the nitrogen base already present by breakdown of nucleic acids or by consumption of food for the biosynthesis of nucleotides. And de novo synthesis means here building a new nucleic acid on ribose sugar. So different molecules will contribute different elements in de novo synthesis of nucleotides. Okay, so these are the references. So this is a brief introduction to the chemistry of nucleic acids. So in your syllabus, so in your next few classes, we'll be discussing this chapter, that is nucleic acid metabolism. So your syllabus is like this: the metabolism of purine and pyrimidine nucleotides. So here we will learn the biosynthesis and degradation of purine and pyrimidines. So there are two pathways for biosynthesis. So both the pathways we will learn. So and then protein synthesis. So that is we will learn both transcription and translation. So then genetic code. So we will learn like how this genetic code is organized and what so the significance of different codes in this genetic code, and inhibition of protein synthesis. So mutation and repair mechanism, DNA replication and DNA repair mechanism. So here we will change the order a bit. So first we will learn metabolism of purine and pyrimidine nucleotides. Then we will go for structure of DNA and DNA replication and DNA repair. Then we will study mutation and repair mechanism. So after that we will study protein synthesis. Then genetic, that is inhibition of protein synthesis, and then genetic code. Okay, so happy learning, have a great day. So next class we will learn and that is biosynthesis of that is the purine nucleotides. Okay, thank you so much.